Whenever an action potential arrives at the terminus of a presynaptic motor neuron, the voltage-gated Ca2 plus channels open. Through these opened voltage-gated Ca2 plus ion channel, the Ca2 plus ions enter into the presynaptic motor neuron. The entry of Ca2 plus ions into the presynaptic motor neuron induces the synaptic vesicle containing acetylcholines to fuse with the plasma membrane of presynaptic motor neuron. Due to this vesicle fusion, the acetylcholines are released into the synaptic cleft in the neuromuscular junction. This neuromuscular junction is a synaptic connection between the terminal end of a presynaptic motor neuron and a muscle cell. The muscle cell's plasma membrane has nicotinic acetylcholine receptor in a closed condition. The nicotinic acetylcholine receptor is a ligand-gated channel. The nicotinic acetylcholine receptors are produced in the brain and are important in learning and memory. Acetylcholine receptor loss is observed in schizophrenia, epilepsy, drug addiction, and Alzheimer's disease. Antibodies against acetylcholine receptors constitute a major part of autoimmune reactivity in myasthenia gravis. Nicotinic acetylcholine receptor is named as such because it is bound by nicotine. Nicotinic acetylcholine receptor is implicated in addiction to nicotine by tobacco smokers. Nicotinic acetylcholine receptor is target of neurotoxin such as conotoxins produced by certain Pacific Ocean snails. The released acetylcholines then bind to the nicotinic acetylcholine receptor. As a result, the nicotinic acetylcholine receptors get opened. At this time, the resting potential of plasma membrane of muscle cell is -85 to -90 MV or millivolt. The Na plus ion present outside the muscle cell and K plus ion present in the cytosol of the muscle cell move simultaneously through the opened nicotinic acetylcholine receptor. Due to this movement, the Na plus ions reach to the cytosol of the muscle cell and the cytosolic K plus ions move to the outside of the muscle cell. The Na plus ion movement is due to Na plus ion electrochemical gradient. Here, the movement of Na plus ion is called as influx and the movement of K plus ion is called as efflux. Due to the movement of both Na plus ions and K plus ions through the opened nicotinic acetylcholine receptor, a localized net depolarization of about -15 MV is produced in the plasma membrane of muscle cell. The muscle cell's plasma membrane has voltage-gated Na plus channels. Due to the localized depolarization of muscle cell's plasma membrane, the voltage-gated Na plus channels get opened. Through this opened voltage-gated Na plus channels, the Na plus ions present outside the muscle cell move into the cytosol of muscle cell. Due to this Na plus ion influx or inward movement, an action potential is generated and conducted. The muscle cells have T-tubules. The T-tubules are specialized invagination of plasma membrane of muscle cell. When the spreading depolarization reaches to the T-tubule, the depolarization is sensed by voltage-gated Ca2 plus channels on the plasma membrane of muscle cell. Through an unknown mechanism, the closed voltage-gated Ca2 plus channels influence Ca2 plus release channel present in the membrane of sarcoplasmic reticulum. As a result, the stored Ca2 plus ions in the lumen of sarcoplasmic reticulum are released into the cytosol of muscle cell through Ca2 plus release channel. Due to the release of Ca2 plus ions into the cytosol of muscle cell, the cytosolic Ca2 plus ion concentration increases. This increased cytosolic Ca2 plus ion concentration induces muscle contraction. One axon terminus of a frog motor neuron may contain 1 million or more synaptic vesicles. Each synaptic vesicle contains 1000 to 10,000 acetylcholines. Please like, subscribe and share.